All right. Good afternoon, everybody. In the meantime, let me remind you that to ask a question to the speaker, you can use the Q&A box on the right side. I think we're all expert of this right at this point. And also, I want to remind you that the posters in the arena are still available for you to kind of browse if you want to. And there's tables in the lounge where you can interact with other attendees. If you go to the poster session, there are some posters that have like videos or PDF links, so check them out, and you can DM the presenter of the poster if you want to meet up and discuss more about their work. Okay, so while people keep joining the session, let me start with the introduction. I'm really happy to have Elsa Bernard here. Elsa obtained her PhD in bioinformatics from the Center for Computational Biology at Mines Paris and Institut Curie in Paris, under the supervision of Jean-Philippe Vert. And she is now a research fellow in computational oncology at Memorial Sloan Kettering Cancer Center in New York. And her work and her interests are on the development and application of statistical and machine learning techniques with a focus on hematologic malignancies. And her current project, which I think is what she's going to talk about today, is in particular related to the molecular characterizations of patients with myelodysplastic syndromes. Is that correct? Yes. Great. So it's on a more personal note, I was very lucky to meet Elsa in Berkeley when she did like a few weeks of visiting in 2012. Was that it? Yeah, when I was a postdoc there.
Wow.
And then I met her again in New York, and I really look forward for her presentation. So without further ado, Elsa, floor is yours.
Thank you so much, David, for the very nice introduction, and also thank you for all the organizers for the opportunity to speak and putting up a great conference. So let me try sharing my screen. Okay, so as David said, my name is Elsa. I'm a research fellow in computational oncology in the lab of Eli Papa Emanuel at Memorial Sloan Kettering Cancer Center in New York. And today I'll be talking about how to leverage large-scale population genomic studies in myelodysplastic syndromes for personalized prognosis. So a word of background so that everyone is on the same stage about myelodysplastic syndromes, or MDS, which are blood cancers. And they're a diverse group of myeloid neoplasms, which are bone marrow failure when the bone marrow doesn't produce enough healthy cells. It's typically a disease of the elderly, with a median age of diagnosis of above 70-year-old. And it's very heterogeneous in the clinical presentation of patients, where lower risk patients can live for a decade with mild cytopenia, while higher risk patients very, very rapidly progress to acute myeloid leukemia, with a very poor prognosis then and the median overall survival of less than a year. There are limited treatment for MDS. The standard of care for higher risk patients are hypomethylating agents, where only half of patients respond, and response is often short lasting. And the only curative treatment is bone marrow transplantation, where clinicians tend to balance the risk of the myeloid disease versus the risk of the transplantation. So in that context, in the clinical workup of MDS, management includes the evaluation of blood counts and bone marrow morphology, as well as conventional cytogenetics, to classify patients into different disease subtypes as per WHO classifications, and also to risk stratify patients into different risk scores as per the International Prognostic Scoring System, where patients are stratified into 5 different categories, with survival ranging from a year to 8 years or so across those categories. So really in the last decades, the evaluation of cytopenia and morphology are driving are the main biomarker for MDS, both for diagnosis and prognosis stratification. More recently, cytogenetics, so with karyotyping, conventional karyotyping, has been implemented in the diagnostic workup of MDS. But only half of patients have chromosomal abnormality, and the other half have normal karyotypes. And with the advances of genomic sequencing and more systematic sequencing at diagnosis, it has been reported, and it's clear that gene mutations are of clinical and prognostic relevance in MDS. However, with the typical long-tail distribution of mutated genes, where most of the genes are most of the recurrently mutated genes are very infrequent, with 80% of mutated genes present in less than 5% of patients. This has challenged the incorporation of gene mutation into formal diagnostic and prognosis schema for MDS. So to formally incorporate gene mutations into classification and risk stratification, we're leading at MSK the International Working Group for the Prognosis of MDS. This is under the aegis of the MDS Foundation, where we analyzing 3,300 or so MDS samples from all over the world, with the main goal to incorporate molecular markers into MDS classification and risk scoring system. So we have received DNA samples from MDS clinics at MSK, 3,300 samples that we've been profiling for genomic alterations. And together with integration of the molecular profiles with detailed clinical data, as well as outcomes follow-up and treatment response, we're working towards a molecular classification of the disease with a biomarker definition and that will inform disease pathogenesis, as well as building prognostic models that are personalized to patients' molecular profiles to provide a more accurate risk estimate per patient and to further guide optimal treatment decisions. So in terms of the assay, of the molecular assay we've been developing and using on those 3,300 samples, it's a simple unmatched targeted panel sequencing assay. So it's unmatched in the sense that we only have the blood or the bone marrow samples and we don't have a matched normal tissue. So this has required some specific variant filtering and QC. So the sequencing panel is comprised of genome-wide common SNPs, so that we can estimate copy number profiles along the genomes. This can be seen as a sparse SNP array, and it has been optimized for regions of LOH in myeloid disease. And the panel comprises also 150 or so myeloid genes that are known to be involved in the pathogenesis of the disease, that we're sequencing with high depths to be able to call subclonal variants down to 2% VAF. And from this assay, we're integrating driver point mutations from the patients, together with conventional cytogenetic abnormalities, such as aneuploidies and trisomies, and a few recurrent fusion genes. And we're also deriving allelic imbalance from NGS data using copy number tools developed specifically for unmatched sequencing data. And so from the integration of mutation, cytogenetic, and allelic imbalances, our first goal is to provide a deep molecular characterization of the disease, a blueprint of de novo MDS, where we find that there are more than 50 genes that are mutated in at least 1% of patients. Patients tend to have three, in average, several driver gene mutations, in average 3. And we identified at least one driver alterations in 95% of patients. And this bar plot is showing you the long tail distribution of recurrent aberration in MDS. We are providing to inform pathogenesis and classification, and I will be talking about classification later on. We're looking into the pattern of commutations, of which genes come together more than expected by chance, or which genes are mutually exclusive with others. And this bar plot is just an example of the interplay between point mutations and co-prenatal LOH, where we see very significant interaction with specific gene mutations and co-prenatal LOH as specific locus in cis, such as TET2 and chromosome 4 and locus 4q, or T53 and locus 17p. And I'll be talking about T53 for quite some time during the talk. And last, we're evaluating the clinical implications of acquired gene mutation in MDS, both for genotype-phenotype relationships and prognosis evaluation. And this Kaplan-Meier curve show you stratification of outcomes for patients with the number of the number of somatic alteration that they have. So from this detailed molecular landscape of the disease, to move forward implementable molecular classification into disease molecularly informed subtypes, as well as prognosis modeling, one of the questions is how to encode features. And gene mutations tend to be encoded as binary, present or absent, and that's usually how they are clinically reported. However, large-scale genomic studies give us the opportunity to define features beyond this binary encoding, taking into account different type of molecular informations. And to do so, we're evaluating genomic alterations at the level of different hotspots or different mutation types, such as truncated mutations or missense mutations, or allelic state as per monoallelic or biallelic. And this lollipot represents G53 gene with a different the distribution of mutations, and you see the different hotspots that are common across pan cancer, and their distribution into monoallelic and multi-hit T53. And in the case of T53, I'll be showing you that the allelic state are of clinical importance for disease for biomarker development in MDS. So this brings me to the outline of the talk, where I first focused specifically on T53 and MDS and how allelic state are important biomarker and should be measured at diagnosis to inform both disease subtypes and prognosis. And then more broadly, we'll talk about how to define molecular subtypes and how interrogate their clinical relevance. And last, our work towards building personalized prognostic models for MDS. So T53 mutations in MDS, so T53, the guardian of the genome and the most frequently mutated tumor suppressor across all types of cancers. And in MDS, it's mutated in approximately 10% of patients, and as in most cancer types, associated with a high-risk disease, high risk of evolution and treatment resistance. So it is strongly associated with so-called complex karyotypes, where in MDS or hematologic malignancies, where chromosome that is high rate of genomic instability. It strongly co-occurs with LOH at 17p, so the T53 locus, leading to biallelic T53. And it's really a marker of high risk disease, with the median overall survival of T53 mutated patients being only a few months. And patients do respond poorly to therapy, and even they are high risk of relapse after bone marrow transplantation. So looking at specifically at T53, we've been integrating mutations at the locus together with allelic imbalances to include deletions or co-prenatal LOH. And here on the bar plot on the left, you see the number of T53 mutations color-coded by the status of the chromosome at the locus. And patients can harbor one mutation or two mutations mainly, and half of the patients with one mutation will have normal karyotype at the locus, or allelic imbalances as per deletion or co-prenatal LOH. And by combining this information, we derived different subgroups that comprise patients with single mutations or multiple mutations, mutation and deletion, and mutation and co-prenatal LOH. So that overall, the main pattern is that 1 in 3 TP53 mutated patients have monoallelic mutations of the gene, here in orange, while 2 in 3, in the different shapes of blue, have multiple hits consistent with biallelic targeting of the gene. And the association between TP53 mutations and complex karyotype and genome instability is very well known. But when we look further at this association across the allelic state, we see a very different pattern, where we can appreciate on the heat map on the left, which represents the main aneuploidies and MDAs together with few trisomies, that the patient with monoallelic mutations have more silent genome karyotype, while the biallelic patients have very unstable genome. So that in aggregate, the so-called complex karyotype, which are defined by having more than 3 chromosomal abnormalities, the association with complex karyotype is specific to the multi-hit or biallelic TP53 state, with 90% of patients having complex karyotype in the biallelic state, while only 10% of patients have complex karyotype in the monoallelic state. So this means that genome stability is maintained. It will perform his gradient of the genome function when one wild-type copy of the gene is retained. In addition to this association with the level of genomic instability, we observe diverse phylogenetic trajectory between the two allelic states, where the patterns of co-mutations are very different between monoallelic and biallelic patients. On the bottom of the list, you can see that the monoallelic TP53 is associated with a broad range of other genes being mutated, very significantly different than the multi-hit TP53, which comes with much less other gene mutations and much less frequent and different. And then by looking at the clonality, so looking at the variant allele frequency of the mutations, we also see we also see higher clonal dominance for biallelic TP53 versus monoallelic, so that the main pattern of trajectory that monoallelic TP53 mutations occur late in disease pathogenesis on the background of diverse other gene mutations, while biallelic TP53 tend to be early clonal events and then strongly associated with complex karyotype. But beyond the genomic association of the TP53 allelic state, of importance is the clinical implications and also outcome and implication for outcomes. So regarding clinical presentation, patients with multi-hit TP53 have much more severe cytopenia. On the top, you see that for hemoglobin, platelet, and neutrophils, where patients in biallelic TP53 are lower than the monoallelic TP53 patients. And they also have increased bone marrow blast compared to monoallelic TP53, and the blast being the stronger morphologic parameter for the assessment for the current assessment of MDS subtypes and risk. And more importantly, TP53 allelic state shapes clinical outcomes, both for overall survival or transformation to acute myeloid leukemia, where you see in the Kaplan-Meier curve on the left a dramatically different outcomes for biallelic versus monoallelic patients, where very surprisingly the outcomes of monoallelic patients is similar to the outcomes of wild-type patients, while biallelic TP53 patients have median overall survival of 7 months or so. And these differences is also strikingly true for progression to AML. And in fact, in multivariate models of survival, so Cox proportional hazard models, TP53 multi-hit is an independent predictor of prognosis along state-of-the-art features, while monoallelic TP53 is not. So this has implications for feature definition and prognosis modeling. And finally, beyond the estimation of risk and prognosis, the TP53 allelic state has implications for treatment response, where we see different survival after different type of therapies, to include hypomethylating agents, lenalidomide, and even with a low number hematopoietic stem cell transplantations. So that one of the conclusions of the study is that the allelic state of TP53 should be considered in future collective studies of treatment response and an important biomarker to incorporate in clinical decisions. So to summarize this first part on the clinical implications of TP53 allelic state in MDS, by integrating mutation and allelic imbalances, we provide a deep molecular characterization of the locus beyond a gene being mutated or not to the level of allelic state. And we show that 2/3 of TP53 patients have biallelic alterations while 1/3 have monoallelic mutations. This has implications for genome stability, where the stability of the genome seems to be maintained when there is residual TP53 in the case of monoallelic mutations. And also, it seems, and this is associated with distinct evolutionary trajectories. And finally, we demonstrate dramatically different outcomes and treatment response for monoallelic and biallelic patients. So this advocates for determination of TP53 allelic state in the diagnostic workup of MDS, which can be routinely implemented using standard sequencing panel assays that combine gene mutation calling as well as copy number evaluation and allelic imbalances. So this first part demonstrates how feature encoding is important for biomarker identification of clinical relevance that can be then incorporated formally into molecular classification of the disease and risk prognosis. So that in the second part, I'll be showing you how we're working toward revising or providing guidelines for the revision of the classification of the disease that might incorporate molecular markers alongside established morphology criteria. So, to study molecular classification, we're using unsupervised clustering analysis, where on the left you see the heat map, just a heat map of the most frequent gene alterations across patients. So patients in columns and genes would be on the rows. And this heat map is ordered by the most frequent gene mutation. And you clearly see that you very quickly lose any structure in that heat map. So we're using we're mostly using some Dirichlet process clustering algorithms that are flexible in terms of the number of clusters to identify robust molecular subgroups that incorporate the commutation patterns and the mutual exclusivity across the different gene mutations in MDS patients. And given those disease subtypes that are then molecularly defined, we're questioning we're questioning the different evolutionary paths of the disease. So how the different pattern of commutations lead to MDS? What is the clonality of the genes within those different classes in terms of order of events? And this should improve our understanding of the pathogenesis. And we're also questioning the clinical relevance of those classes. How do they align with established disease subtypes, which are defined by morphology criteria mainly? What are the clinical phenotypes across those different subtypes, as well as the different outcomes and treatment response across those molecularly defined entities? So that the heat map on the left will represent a more detailed heat map of the classes we identify. So we do identify quite a number of classes in MDS, given the high genomic heterogeneity of the disease. And for each one of those classes, we're using some statistical model called Bradley-Terry model that are borrowed from sports statistics and look at the different all the pairwise comparison of gene mutations per patient in order to estimate which gene mutations comes first and which gene mutation comes second, clonal versus subclonal, and then from those estimate a relative ranking of the gene mutations per class. So here you see, for instance, the clonality of the different genes, where some genes have variant allele clonality adjusted for copy number. So some gene mutation will have variant allele frequency, adjusted variant allele frequency around 50%, so they tend to be more clonal, and some other will have more subclonal peaks, so they tend to be secondary acquired mutations on a already existing background. And broad letter remodeling that have been extensively used in previously and more recently also in the Peacock papers provide a relative ranking of those events across time. And so this inform of the different evolutionary trajectory of the disease that can be very diverse and that differs across the molecular classes. And then to investigate the clinical relevance of the classes, the first question is what is already recognized and what is new. And this bar plot simply shows the number of classes that we identify. So provisionally, we have 17 molecularly defined classes. We have 5% of patients that do not have any detectable driver alterations, and then 8% or so that are not positioned within a molecular subtypes. But then from the other classes that ranges in size for only a few percent of patients to 15% of patients, some of the classes really align with established WHO classes as per their strong genotype to phenotype or genotype to morphology correlations. Some others are recognized classes of already recognized genomic alteration that have been recognized by previous literature, such as typically complex karyotype. But most of the others are new classes that involved a high number of gene mutations, 3 to 4 in average, and are defined based on the pattern of commutation and mutual exclusivity. And so, importantly, those classes have clinical relevance, both in terms of clinical presentation and outcomes. And here you see the variation of blast across the molecular classes, where the lower risk classes have very low percentage of bone marrow blasts. So the bone marrow blasts are those immature cells in the bone marrow, while the higher risk classes have quite a higher median percentage of blast, with some of the patients have close to 20% blast, which is a threshold for AML for diagnosis of acute myeloid leukemia. And looking at the outcomes across those classes, also very strongly stratified patients from patients with median overall survival of more than 7 years to months. And then the last example of importance and clinical utility of molecularly defined disease subtypes I would like to give you is by looking at a specific example, which is SF3B1 mutated MDS. So SF3B1 is a splicing factors. It is one of the most commonly mutated genes in the disease, and it's been recognized. It's one of the only molecular marker that is already incorporated into the classification. And it has been recently proposed that SF3B1 mutated MDS is a distinct subtype, both because of its mechanism of actions as well as clinical implications. And typically, SF3B1 disease is a favorable disease with milder phenotype and longer survival. And when and the bar plot on the top shows again just the size of the molecular classes color-coded by the presence or absence of SF3B1, and presence is blue. And we clearly identify one class which is driven by SF3B1 only, and which is in line with the recently suggested WHO International Working Group recommendation for SF3B1 disease. But we also have some other classes which are enriched for SF3B1, and those classes then do behave quite differently. And in particular, so the commutation pattern are very different in those other classes, which is not the typical SF3B1 disease class. And when we look at the outcome across those classes, we clearly see that in those other classes that the SF3B1 class, SF3B1 is not conferring a good prognosis. There is even one class where SF3B1 is conferring a less favorable prognosis compared to the average of the class. So this really illustrates that molecular context matters, both for disease pathogenesis as well as prognosis. And so this has implications for feature definition and prognosis modeling that extend beyond single gene biomarkers. So this illustration of how genetic context is important for prognosis brings me to the last part of the talk, which is our work towards building personalized prognosis models for MDS patients that incorporate molecular markers as well as established criteria. To develop those prognosis models, briefly in terms of methodology, which is state of the art, we're doing it across different endpoints that include overall survival or AML-free survival, as well as AML transformation. Feature definition, I've been talking about that, include state-of-the-art clinical features, blood counts, blast, and cytogenetics, as well as gene mutations encoded and as binary gene mutations, or at the level of hotspot, or at the level of monoallelic versus biallelic states, together with novel biomarkers that include congenital LOH or others. And we're testing different algorithms on the cross-validation framework and assessing performances with survival-specific measures such as C-index. But beyond this evaluation of predictive performance, the predictive ability, we're also studying the dynamics of the predictive ability and how much of short-term versus long-term are we able to predict. So, I probably don't need to say that to this audience, but survival analysis can be seen as a time-dependent classification where conditioning on the censoring of patients. And this time-dependent classification can be evaluated with time-dependent ROC curves, for example, where we look at the IUC across time. And typical integrative measures that are used to evaluate prognosis models include C-index, which is just a weighted average of the time-dependent AUC, weighted by the inverse probability of censoring. And we investigated C-index across different type of models that are only based on clinical features or only based on molecular features, towards more integrative models that include the combination of features, and we see very significant improvement in terms of C-index. But beyond these integrative measures, we also look at the predictive performance across time, and this will be a representation of the AUC over time for overall survival and AML transformations for different type of models that are only the demographics or only clinical or molecular or more integrative measures. And we see here independent predictive power of both clinical and molecular features over time, where the AUC over time is maintained from 75 to 65 for overall survival, for instance. So there is a drop of predictability over time using molecular and clinical features for overall survival, a drop which is less so existent in AML transformation. And we see here, for instance, that how different endpoints behave differently, where age, as expected, is predictive for survival. We're talking about the elderly population, but it only becomes predictive as 0.5 being a random prediction after a few years, so that really the short-term predictions are driven by the clinical and molecular profiles, and on the longer term, the predictions are more driven with other factors that include the demographics. While for AML transformation, the age is not predictive, and the most predictive features are the molecular features, so that the integrative models have fairly constant 0.8 AUC over time. So to conclude this part of personalized prognosis and patient-tailored risk estimates, we're working toward implementing a web-based calculator that will be hosted by the MDS Foundation, which already have an app that where blood count features, hemoglobin, neutrophil, platelet, as well as cytogenetics, and we're overlaying this with molecular features to provide a specific risk estimate per patient that include their molecular profile, both for overall survival and AML transformation. And so with that, I would like to thank, first of all the collaborators that provided samples, materials, as well as clinical data. My mentor in that project, Ali Papa Manuel, together with a member of the lab, different structure at MSK to include the sequencing core, cytogenetic core, and biostatistician people, other collaborators from the study, and as well as the WHO committee and the risk IPH prognostic international working group for the prognosis committee that are also providing guidance. And I would like to thank you for your attention, and I'm very happy if there is any question.
Thank you so much, Elsa, for the fantastic work and a great presentation. I already see a few questions in the Q&A tab, so I will just start with those. So Johan How, okay, I don't know how to pronounce his last name, but would it make sense to include epigenomics data such as DNA methylation markers, or have you by chance tried including such data in the prediction models?
So I haven't tried because I don't have this data, especially at this scale. It might make sense, especially for predictive questions of response to HMA. Because the state-of-the-art treatment is hypomethylic agent. So and there have been reports of predictive ability of methylation marker for response to HMA. So definitely, but I think what would be interesting is to use the molecularly defined subtypes and overlay the methylation marks and the transcriptomic on those DNA defined subtypes. Great question.
Yeah. So we have another question by Wolfgang Huber. Did you check for the presence of chromothripsis in your samples? For example, did it coincide with the multi-allelic p53 mutations?
Mm-hmm. It's a little bit difficult because this is a panel. This is not whole genome, so it's a bit hard to infer chromothripsis from panel sequencing data. And also, chromothripsis is not very frequent in whole exome studies of AML and MDS. So I cannot answer for sure, but clearly so I don't know. We haven't been able to map chromothripsis with targeted, but clearly some genomes that are the most complex with almost all chromosomes being altered are clearly associated with B allelic TP53. But we cannot call that it's not called chromothripsis.
So Alejandro Reyes asks, are the associations between allelic state of p53 and other mutational patterns specific for AML, or would you also see it in different tumor types? A pan-cancer thing. I also was wondering the same thing.
Yeah, so that's a great question, and that's of course one of the immediate question we had after doing the MDS study, and we're actually looking, but this is like very recent work. We're looking at the pan-cancer data set to evaluate this question across different tumor types. Also, I must say, from first pass evaluation of the data, solid tumors are a very different beast, and they have a high level of whole genome duplication, while whole genome duplication is very infrequent in AML or MDS. And the presence of whole genome duplication is a fertile ground for genomic instability, and the picture might typically state might matter, but the implication will be fairly different.
Okay, so we have a question by Richard Salisbury. Very nice demonstration of the importance of not treating mutations as purely binary in p53. Have you also done this for other mutations, and how easy it is to integrate into your model predicting clinical outcomes?
Mm-hmm. Yeah, it's a good question. Indeed, I focus my talk on TP53 because the implications were the most important in terms of prognosis. But we have done it systematically where it was possible to do. That is where genes harbor multiple type of alterations, and we do see other cases where binary encoding is not capturing the entire informations, such as some genes have different hotspots, and those hotspots have different pattern of commutation from one hotspot to another within the same genes, and this is true, for instance, for IDH2. And it's not fairly once you have mapped this pattern out of the data is not fairly difficult to implement in the prognosis model, because we have the information from the sequencing assay. We know which hotspot are mutated, we know the clonality, and provided that you also have a copy number to evaluate LOH, you can fairly easily estimate, for instance, monolithic versus biallelic. Yeah.
Great. So there's another question by Wolfgang Huber. How would you see the path towards clinical testing of the personalized risk predictor and its efficacy for treatment?
Well, so I'm not sure what you mean by clinical testing. For sure, there is committees supervising the revision of both the WHO and the risk scoring system, and those committee integrates statisticians as well as clinicians, so that we have to work to speak together and reach a consensus in terms of algorithmic versus interpretation. And I think we're working toward an interpretable model where that will also help clinicians to understand what is going on, but also, and the web app will be more to be able to even show patients where their molecular profile fall into the MDS landscape. So there is both a statistical work as well as educational and knowledge transfer path. And regarding treatment, this will require more prospective studies that specifically integrate for instance, the molecular class information.
Okay, so I think we're at the end of the session. So in the interest of time, I think if you you have more questions for Elsa, perhaps she can stay around a little more. I know that we can give an extra thank you to Elsa, because I know she told me it's snowing in New York and there's no childcare, and so a big thank you for giving the talk anyway.
Yeah, but my daughter was not interested in coming listening, so everything went smoothly.
All right. So, and if she can stay around, please find her in the lounge and maybe ask a further question there.
Yeah, that would be great.
Yeah. Thank you again, Elsa.